Hello and welcome to everybody. This is our fourth session of the SQAB tutorial presentations that ABAI kindly hosts. And we have a winner to bring us home with these SQAB tutorials. The president-elect of SQAB is going to talk to us, Dr. Fed Sanabria, and if you look at page 125 of your program, you will be able to read his illustrious credentials. I've been told that he wants to have time for questions, so I should not go on too much, but suffice it to say that Fed had got his PhD from SUNY Stony Brook with Howard Radcliffe. He then moved to Arizona State University and worked with Peter Killeen. He's done seminal work in timing and the structure of behavior, and he is going to educate all of us today about bouts of behavior and how to define bouts and understand them. So with no more ado, please let's give Fed a warm ABBA welcome, ABAI welcome.
Thank you, Suzanne, for that very kind introduction, and thank you everyone for being here. Yes, I'm going to talk about bouts of responses, but before I do that, I want to acknowledge that a lot of the data that I'm going to be showing you and a lot of the ideas that I'm going to be sharing with you came from, a bunch of other people. I want to acknowledge my current student, Matthew Gilday, and former graduate students. Much of their data is actually I'm going to be showing to you. Ryan Brackney, Corda Daniels, Tanya Gupta, a former postdoc, Christina Santos, and some collaborators. Also, funding source, and there's a name that I'll never include here because it seems it doesn't belong with the rest of these names, which is Peter Killeen, whose ideas I keep stealing and making them worse, I suppose. I changed them. I tried to make them myself. I'm like the ChatGPT of Peter's ideas. All right, so quick outline, what I'm going to be talking about. Responsive bouts, what are they? Why are they important? How are they produced? How we analyze them? And I'm going to split this into two parts, visualization and parameter estimation, which is going to be real quick. And by the way, the parameter estimation part, I'm so glad that I went after Greg because he did more than half the job that I'm going to be showing you, so I have even more time. And I'm going to talk about some constraints of this way, of this approach, and its generalizability. And for that, I'm going to be using the behavior of a man called Elon Musk. You may know him. And if you want to know what is it that I'm going to use about his behavior, you're going to have to stay till the end of the tutorial. All right. So this is interesting. It seems like we all think about lever presses as vertical ticks, and time as a horizontal line. That's how Greg was presenting some of his examples. That's how I'm presenting it. So this is how data, for rat as a lever for food or any kind of discrete behavioral event may look like. You have a bunch of responses, and they may be organized, and they're typically organized in kind of clusters over time. And they're relatively long pulses between clusters of individual responses that are separated by relatively short pulses. So if this is the case, if this is true, if indeed behavior is organized this way, at least under some circumstances, and those circumstances are typically particularly visible, this pattern of behavior in variable interval and ratio schedules, then characterizing this pattern of performance could be, may rely on just identifying a few parameters. So those are, the characteristics of the bout lengths. They're, some measure of central tendency, some measure of their dispersion. Also the characteristics of parameters that describe the distribution of inter-bout intervals, that is the intervals between that separate consecutive bouts. This would be shown as relatively long inter-response times. And finally, the distribution of those intervals that separate responses within a bout, which should be relatively short IRTs, short inter-response times. If you have all this information about, each of these three aspects of this behavioral data, you may know everything that you need to know about those data. And now, when you break up this data that way, looking at bouts and looking at the length of these bouts and so on and so forth, you're not really losing any information, right? Because you can compute, if you're really into, using response rate as your dependent measure, you know that your the response rate is simply the reciprocal of the mean IRT. But if you can recover the response rate by simply having, using that equation, let's see if this is probably easier. Using this equation in which you have the mean length length of the bout and so on and so forth, divided this way, you got response rate. You're not losing any information by breaking up, by analyzing behavior in this particular way. So that's great. Now, why should we care about this, right? And the reason why, at least I care about this, is because it appears that each of those parameters that I identified seems to be selectively sensitive to very particular manipulations. So, for instance, in the case of the mean of the inter-bout interval, it seems to be selectively sensitive to changes in rate of reinforcement, magnitude of reinforcement, response effort, well, not response effort, I'll tell you a little bit more about that. It's sensitive to response effort, but not selectively. Deprivation level, response required, a number of different things that we care about. Response effort, in fact, it's kind of a tricky manipulation because it also appears to change the intervals between consecutive responses within a bout. And the length of these bouts appears to be selectively sensitive to what we ask the subjects to do to collect reinforcement and to the presence or absence of non-contingent reinforcement. And so, it appears, at least, that each of these three parameters, the key parameters that describe the structure of behavior, the distribution of behavior over time, are selectively sensitive to three important aspects of behavior. Or three, to the extent that, B is selectively, this parameter B is selectively sensitive to motivational manipulations, it seems to be indexing this underlying process. Motor processes seem to be tagged to, or W seem to tag these motor processes, and the schedule effects seem to selectively affect the length of these bouts. So this is analyzing behavior. This is taking the performance of a subject, and this seems to be allowing us, if we know these parameters, it may allow us to draw some inferences about underlying processes that are not readily evident by just looking at those vertical ticks on a horizontal line that I just showed you. But there's more. There's more to this, I think. I think it raises some questions about what is it that reinforcement reinforces? Because you may think about an added reinforcer as a piece of candy. You can think that that piece of candy is operating on the response that actually produced the reinforcer, or maybe it operates on bouts of activity. It seems like the latter is probably a better description based on some data. What does extinction extinguish? So this is data actually that Ryan Brackney in my lab collected some time ago, in which he simply trained animals on what I believe it was a VI 120 seconds with a tandem requirement of 5 responses. And what I'm showing you here is the IRTs that the animals produced on a log scale as a function of time in extinction. And hopefully you can see that you have two very clear separate populations of inter-response times. One that is very insensitive to extinction, and that's very short. These are your within bound intervals. And another set of another population of IRTs that is much longer, that is sensitive to extinction, and it's the one that separates bouts, presumably. So what is extinction extinguishing? It's not extinguishing simply responses, it's extinguishing the likelihood that a bout of responding would be initiated. We've asked other questions, like it raises other questions. Are all forms of response disruptions, like the ones that are used or were used for testing, say, behavioral momentum, functionally identical? Is extinction ultimately functionally the same as the devaluation of the reinforcer or the introduction of non-contingent reinforcement? And the short answer is no. With Brian Bragg, they showed that these are clearly dissociable. And overall, this whole thing just shows that there is a misalignment, a surprising, at least to me, somewhat misalignment of behavior with environment. If you compare this, for instance, with a matching law, right, that shows you that behavior is attuned to the distribution of reinforcers, and the behavior is distributed according to how reinforcers are distributed. Well, if you think of that, how that should happen over time, if reinforcers say on a variable interval schedule, when the probability of a reinforcer is roughly constant, you would expect that behavior will be produced at a constant rate. Yet that's not what happens, because the probability of getting a response, given that response was just made, is higher than if the animal had not made a response. And so that seems to go counter to some intuitions about how behavior adapts to the environment. And another point is really trying to highlight why I think this is important, is that I think that overall it suggests a framework for investigated motivated behavior that is somewhat different from the way that we traditionally think about motivated behavior. And if you want to know the details about that, you can either read that paper, or you can come to me and be more than happy. And I'd be happy to tell you about it. After 30 minutes, you may not be as happy as I would be. But I have so much to say about this. Anyways, and there's a lot of other reasons, I think, why this, I think, is important. So hopefully I've persuaded you that at least the idea that behavior is organized in bouts over time is at least not unimportant. But how are they produced? Where do we see them? In the laboratory, where we typically produce these kinds of data, we've seen them in human and non-human animals. Phil Reed has done a number of studies showing that human behavior in the laboratory is organized in bouts. And Rick Scholl in the early 2000s conducted a number of seminal studies with rats showing that, in fact, this is the way that their behavior is distributed. What they use, and what many of us have used typically, is variable ratio or variable interval schedules of reinforcement. The reason for that is because these schedules generally produce, if you arrange them in the right way, they produce inter-reinforcer intervals that are exponentially distributed. That is, that they look as if they are being produced at a relatively constant rate. And so the intuition is that if reinforcement is produced at approximately a constant rate, behavior should also, correspondingly be emitted at an approximately constant rate. And that's not what happens. Interestingly, Takeyuki Tano, some years ago, replicated a classic study from Charlie Catania in the late 70s, trying to show that comparing variable ratio and variable interval performance by yoking reinforcement relative to the other, so that the pattern and the rate of reinforcement is identical in both schedules. Charlie showed back then that on a ratio schedule you get a lot more responding than on an interval schedule. Tano replicated those data, but he looked at the structure of the behavior that was produced, and he shows that the key difference in performance between those two schedules is the length of the bouts that are produced. Variable interval schedules produce really short bouts. Variable ratio schedules produce much longer bouts. So this is giving us some ideas about where these differences are coming from. But for the most part, this is the kind of schedule that typically produces the kind of data that you will see that I'm going to be showing you. So this is what I mentioned now. And oftentimes, a tandem ratio requirement is appended at the end of a VI schedule. And the reason for that is that it appears to make these bouts of behavior more visible. So if you train, like I showed you data from Ryan Brackney, an animal in a VI 120 seconds with a tandem FR5, what you see is that you see bouts of responding that on average are 5 responses long. Which is interesting because there is nothing signaling to the animal that they have to make 5 responses to obtain reinforcement. They get it just through feedback. All right, so this is how we typically produce this kind of data. So next thing that I want to show you to discuss is how are these data analyzed. So you have something that looks like this. Is this 1 bout? Is this 2 bouts? How do we decide between those 2 possibilities? Well, that question is really the question about what is this interval here in the middle? Is this a within bout interval? And in that case, you have 1 bout. Is this one a between bout interval? Then you have, this is the interval that separates the 2 bouts, right? And so how do we think about those intervals? How do we tell them apart? Well, we can't for sure with a high degree of certainty, but we can assume that we are sampling these intervals from 2 distributions of inter-response times. One distribution of relatively long inter-response intervals that corresponds to the population of inter-bout intervals, and another one of much shorter inter-response times that corresponds to the population of within-bout intervals. And there is some probability that these intervals are going to be sampled from one distribution, and a complementary probability that this is going to be sampled from the other distribution. And so what we need to do to tell whether the extent to which what we're looking at here is 1 bout or 2 bouts or whatever, we need to estimate the parameters of this mixture distribution, of the sum of these 2 distributions. So that's how we analyze data. And so essentially, well, the first thing that I want to clarify is that even though I use this to signify distribution, this kind of like bell-shaped distribution, actually the distribution of IRTs are very, very skewed. In fact, most of the times they're exponential. An exponential distribution does a good job at describing both the distribution of long and short IRTs. Such that if you plot this distribution in a semi-log plot, you get straight lines. And straight lines are always your best friends when you're analyzing data. Because then, if you take the slope of these straight lines, you get everything that you need to know about these distributions. You only have 1 parameter for each of these distributions, which is, the reciprocal of the slope, the negative of the reciprocal of the slope. That's your mean and your average, your mean and your standard deviation of the IRTs in that distribution. And lo and behold, something really interesting, something that makes life a lot easier is, remember that we got a probability of sampling from one distribution to the other. Well, if you take the reciprocal of that probability, the probability of sampling from the long IRTs, that's the average length of a bout. So let me illustrate that here in terms of, this is sort of like the general rationale, how you go about estimating these parameters and why you want to estimate these parameters. Let me show you how you want to visualize this process. So again, we have this behavioral data, and now let's suppose that you knew which particular intervals separate bouts. If they are indeed exponentially distributed, then you would expect that a semi-log survival plot, like the one that I'm showing you here, will give you a straight line that looks like this. Now, what this plot is plotting is the probability that you will have an interval greater than whatever is on the x-axis. So, what is the probability that you will have an interbout interval greater than 0 seconds? Well, all of them will have to be greater than 0 seconds. That's why this probability is 1. And as you change, you move towards the right on the x-axis, of course that probability goes down, 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 down. And if it's an exponential distribution, and you take the y-axis and plot it on a log scale, you get a nice straight line. So that's how you would know that these intervals are exponentially distributed. Same thing happens with bout intervals. Much shorter, of course. So now, whereas before, let's say on this example, interbout intervals that were greater than 5 seconds were about, I don't know, the likelihood that an interbout interval will be longer than 5 seconds is only 0.2. It will be essentially 0 when we're looking at, in this example, when we're looking at within bout intervals, right? They are way, way shorter. That's why this distribution, this survival plot, is much, much steeper. Okay. The cool thing is, of course, you don't know which ones are what. You don't know which ones are separating bouts. You don't know which ones are within a bout. You see the sum of both. And when you add both of them, you get this nice, you expect it to get this very nice broken stick pattern, right? The sum of the two distributions. And the shape of this survival plot has very specific meaning. The slope of the shorter part corresponds to the within bout response rate. How fast is the individual responding when in a bout? And the longer part corresponds to the bout initiation rate. How often are bouts initiated? And if you project this line all the way to the y-axis, you look at the y-axis intercept of that projection, you get the reciprocal of the length of a bout, measured in number of responses. So, got all the information that you need, presumably, in this plot. That's why oftentimes folks that try to analyze the data this way, or take this approach, oftentimes plot this semi-log survival plots. They're very convenient. Of course, the next question is, does the data look like this? And of course it does. I wouldn't be here if it didn't. This is data from one animal from Ryan Brackney, again, from 2011. And so this seems to work really well. Now, the next step, of course, is, now that you know how to visualize this, is, well, how do we go about, well, how do we implement this? How do we implement this visualization, right? And so you can do this in a number of different, computational statistical packages, all sorts of fancy things, or you can do it in Excel, your choice. So what I have here, and I know it's a little bit small, so I'm going to try to make it a little bit, more visible for you, is that what I have here is the typical output from any of these experiments. So you have time tags for responses, right? This indicates at what second each response happened. And what you want to get from, what you want to extract from this data are the IRTs, the inter-response times. which is simply the differences between consecutive time tags. Nothing too complicated, hopefully, right? And then what you do is you rank those IRTs in descending order, so that an IRT that is really short, like this one is 0.01, is ranked really high, and one that is really long. This, by the way, is actual data from Angel Jimenez. We did this work with him. This is an interim response time of 18 seconds. It's ranked really low, 42, right, out of 1,066 IRTs. I think these were animals trained on a VI 120 seconds also. Then you divide that rank by the number of observations, or 1,066, and now what you have is essentially the y-axis in the graph that I showed you before, the probability that you're going to get IRTs longer than the one that you just observed. And you plot that. But of course, I plotted it here on a linear scale for the y-axis. Doesn't look that great. Looks a lot better when you log transform that y-axis, and here you have your broken stick. This portion of the stick, of course, the short one, is much shorter than the one that I showed you from Ryan, mostly because there was no tandem requirement in this example. So that's, again, why the tandem requirement also often makes this Bouts more visible. All right, so that's how if you want to visualize this data, that's, if you don't need to bend your data, you need to average, you don't need to transform it, just throw it in something like this, and you can get, you can visualize that broken stick, or at least you can visualize the distribution of your data. So now, next part that has to do with parameter estimation. So now you visualize it. How do you know, what are your parameters? What's your best guess? What is parameter B? What's parameter L? How do you go about doing that? And for that, you need to have, we're turning this intuition into a simple equation. And this is just saying that the probability of an RT is the mixture of two probability distributions, two exponential distributions. One that has a mean of B and one that has a mean of W. With, one with probability B, one with the complement. And remember, that probability P, that an inter-response time is going to be sampled from the interval distribution, is just 1 over the length of a bout. And I was smiling when Greg was talking about maximum likelihood estimates, because that's exactly what I'm talking about here. So you can go about estimating parameters by using the method of maximum likelihood. There's a number of methods. In fact, Rick Schultz suggests a different one. I think the most principal one is this, because what it does is, by varying these parameters, parameters that we care about, we're trying to maximize this probability. That's essentially it. And then we get pretty close to predictions from the model that fit really well to the data. So how do you go about this? Fairly straightforward. This is the IRTs. Remember that we obtained by taking the differences between consecutive time tags, but now they're ranked already, right? Here are the ranks, in descending order. Rank over n, going from 1 all the way to a really tiny number, very close to 0. And now, using the equation that I show you, you can actually, not that equation, I'll show you the equation that we use in this case. We want to produce the predictions from the model. And so we have the 3 parameters from the model. These are just initial estimates, just guesses. And what you do is you say, well, if this were the real parameters, what would the data look like? But in this case, notice that we're looking at, what we want to estimate is what is the probability that there's going to, that we would observe an IRT that is greater than each of these values, right? And this is the equation. It's not a lot different from the previous one that I showed you. This is just a cumulative distribution function from the previous one. So you do that, and you plot those predictions against your data, and then you adjust those parameters manually to make it sort of fit, and you're probably pretty close already to the parameter estimates, to the maximally likely parameter estimates. Oh, I forgot, I skipped one step. You want to compute the likelihood of those parameters. So you want to compute what is the probability that given these parameters, you will observe this particular IRT. And this is the equation that we would use. These are really probability densities, not real probabilities of when you get numbers above 1. But that's okay. You take the log of those likelihoods, you add them, remember Greg explained a little bit of this, and you get what is the probability of all your data given these parameters. How likely it is that you would get, how likely your model is, that what is the probability you will get all those data given that those are the parameters that are actually operating. If you're familiar with Solver, Excel has a Solver add-in that if you don't use it, you should. And you can use it to maximize the log likelihood. And when you do that, you get your maximally likely estimates, and that's it. You get a set of numbers. Now, and you can see the fit is not a whole lot better than my guess, because this is the beauty of straight lines. You can pretty much by hand, fit a straight line to data that is distributed in a kind of like straight line. Now, what about those numbers? So let me make sure that we're clear about what those numbers mean, how we interpret them. So here's our parameter B, was 7.39 seconds. And one thing that I think that for whatever reason, I'm going to use this opportunity to emphasize this, for whatever reason, behavioral analysts are terrible with units. We don't like it. People talk about K parameter for delay discounting. Nobody puts units on them. It has units. So always please use units. So B has units. It's seconds because it's an interval. It has seconds, minutes, hours. Right? In this particular case, because we measure the IRTs in seconds, B has 8 seconds units. So the mean time between bouts was 7.39 seconds. You want to represent that in terms of rate? Sure, take the reciprocal. That's 8.12 bouts per minute. So change it to minutes so I will get something like, a fraction of a bout per second. So at least you can start visualizing what the animal is doing. Every 7.5 seconds or so, the animal initiates a bout, right? And when it initiates a bout, it produces responses every 0.38 seconds. That is 2.66 responses per second, which seems reasonable. This is a rat pressing the lever about 2 or 3 times per second. It seems like a reasonable rate, right? And it produces very few responses though, about 2 in this particular case, right? I think this gives us a much more complete picture of what is the animal doing than just saying, that the animal was producing 14.81 responses per minute. Particularly if we know that there are manipulations that selectively affect each of these different parameters. There are more tools that we've developed that I'd be happy to share with you if you're interested. I'm just going to mention them here. What about the distribution of bout lengths? Turns out that distribution depends on what we ask the animal to do to collect reinforcement, and we figure out ways in which the distribution of those bout lengths can be estimated. And in fact, by doing that, we discovered that there is a bunch of really short bouts, just one response, that seem to contaminate all our data. And what seems to be happening is a little bit speculative, is that even when we ask the animals to collect food by making 5 responses to get food, sometimes they just make one isolated response. My guess is that response is functionally different from the other 5 responses that were probably controlled by its consequences. That one isolated response The animal just bumped into the lever. I don't know, was doing something else, was exploring, doing something that had nothing to do with reinforcement. And there are ways in which we can actually look at the prevalence of those disengaged, we call it disengaged responses. Those are analytical tools, using the kind of data that I've showed you. But we've also developed some empirical tools. Using revealed operant techniques with Carter Daniel some years ago, we figure out ways in which we can tag the initiation of a bout and separately tag with a different response the continuation of that bout. So that way, all this trickery wouldn't be necessary, right? And we show that, in fact, this works relatively well. But of course, that requires a very specific setup for having separate measures of these two important variables. And oftentimes, particularly natural data is not organized that way. Which leads me to talking about some of the constraints and the generalizability of some of the things that I've talked about here. The main constraint, I think, is you need tons of data to do this. If you have very little data, your parameter estimates are not going to be that great and not very reliable. However, unlike situation many years ago, nowadays we can collect tons of data, natural data in fact, from humans, or other species for that matter, through a number of devices that are available today, like smart devices, activity trackers, and even social media, right? So we're in a world full of data, so that's good for this approach. And an important thing is that even if, as it's the case in applied settings in particular, if you're working with relatively small behavioral samples, it's always good, I think, to be aware that the principles that operate on large behavioral samples probably also operate on small behavioral samples. So perhaps being aware that a response, whether it's initiation of a bout or the continuation of a bout of activity, are sensitive to different aspects of the environment is probably something that is important even if you are working with relatively small behavioral samples. Now, as I promised, I want to show you some natural data. This is actually the tweeting behavior of Elon Musk between mid-February and mid-May 2023. I was thinking about, well, where can I get free data publicly available from a public figure, presumably, that I can analyze this way. And well, you can see this individual produced 3,114 tweets in a matter of months. So it's great. It's better than any of my rats. So what we did is we plotted the proportion of inter-tweet intervals. We did that semi-log survival plot, and that's the distribution. Looks orderly, at least. Looks nice. So we said, all right, let's look at that behavior. And so this is what I can tell you about Mr. Musk's tweeting behavior. I have an asterisk. I'll get back to the asterisk in a little bit. So the mean bout initiation rate was about one bout of tweeting, a little bit over about every hour. I took out intervals longer than 4 hours, assuming that the man is sleeping or actually doing some work. The mean bout length is relatively long, almost 3 tweets. So every hour, on average, he will sit down and on average produce about 3 tweets. And those 3 tweets will be separated by about 5 minutes. I think that tells me a lot more about his behavior than just how many tweets per hour he produced on average over the course of 3 months. Now, here's the downside. That's a reason for the asterisk. Don't trust those data that much. The fit is not that great. Bummer. I thought that this would fit a lot better. And so what's going on? Well, first of all, don't trust those numbers that much. So one possibility is perhaps, I've been talking about only one type of, or two types of pauses between responses, right? Long ones, separate bouts, small ones within a bout. Maybe there's more than one type. And some folks have used that approach to fit data like this. The problem with that is that, where are all these different kinds of pauses coming from? It's a little less intuitive the more kinds of pauses you have. Maybe it's a completely different mechanism that is producing this kind of presumably reinforced behavior. We don't know. In any case, in summary, what I hope that you take from today is that instrumental behavior is organized in bouts, even when the reinforcers are not, and that this is important. You can easily produce this in the lab. I think that there's a lot of natural data that is organized this way, but in any case, if you want to implement this approach, do it with lots of data. Hopefully, you walk out of this tutorial today knowing or having a good idea about how to visualize this data and how to estimate parameters. And of course, if you get stuck, you can always email me. And there's clearly still more work to be done to account for natural behavior. But at least we know that there is that we know what are the limitations of this model, of this approach when we try to apply it to natural data, natural behavior data. And that's all I have. Thank you very much.